All right, we'll move to our second talk. Our second speaker is Eric Aguiar from Brown University. He's in Soren Israel's lab. And Derek is going to speak to us about DELISHUS, an efficient and exact algorithm for genome-wide detection of deletion polymorphism in autism. Derek.
Thank you very much for that. Yes, this is an algorithm for detecting deletion polymorphism in SNP data, and it's inspired by genomics of autism. This is a joint work with Bjarni Halldórsson from Reykjavik University, Erik Morrow from Brown University, and Soren Israel from Brown. So there's a growing body of evidence that suggests genomic deletions play a major component in disorders of cognitive development. And this is especially true in the case of autism, where I've just shown a sample of papers here. There are many other examples where these research groups have implicated deletions in genes that seem to be associated to the autism phenotype. However, most of these deletions that have been found have been large and very rare. Like with most complex disease, much of the heritability of autism is yet to be explained. So our goal is to develop an algorithm, a set of algorithms that are capable of inferring small recurrent deletion variation in genome-wide data. So this type of variation really hasn't been probed too much in these genome-wide association study data sets because it's very difficult to find the smaller deletions. So before we get into the method, there's three main types of experimental methodology for calling deletions. The first is intensity-based. And what we do here is we can look for contiguous sets of probes that have low intensity or high intensity, and these would indicate either a deletion or a duplication respectively. So there are fine tiling arrays that have very short distances between probes in which you can infer small deletions very well, but it's very difficult to apply these genome-wide, and especially to large cohorts that you would find in GWAS data. You can also do the same type of methodology and algorithms on SNP chips. And they do extend genome-wide for many individuals, but they have much lower resolution than the fine tiling arrays, and they have difficulties finding small deletions because you need a lot of probes, and the intensity signal is very noisy. These algorithms are also very computationally expensive to do for a GWAS data set. The second deletion calling method is read mapping based. So this uses a combination of read depth and reference sequence mapping. For example, you sequence a patient's genome, align it to the reference, and then regions that have little or no mapping correspond to potential deleted sites. Now these can accurately infer deletions and deletion breakpoints down to base pair resolution, but there are some problems. There are high false positive rates from regions that cannot be sequenced, or problems with the mapping. Biases do exist from the choice of reference assemblies, so different reference assemblies will give you different sets of deletions. There's also biases from non-random sampling of the genome during the sequencing process, and this is still very expensive to apply to GWAS type data. The third set of methods is the family-based, or the family-based deletion calling from SNPs, or technology neutral. All of these methods rely on the fact that if an individual is hemizygous, so has a deleted haplotype, and what I mean by that is just a set of SNPs, so a piece of one chromosome, a deletion haplotype and a normal haplotype, then that individual will be called homozygous for the interval of the deletion. And this is just an example with the blue SNP. The individual should be heterozygous, but due to the deletion haplotype, either the sequencing method will only sample from one haplotype, or the intensity-based methods will only see one intensity from the A allele, and the individual will be called for the allele that's present. So for dense SNP arrays, this has the potential to infer small deletions, but it too has some caveats. It can suffer from high false positive rates due to badly typed SNPs. So you have to control for this. It has less power to infer sex chromosome and de novo deletions. And also, the resolution is limited to the type SNPs, so you need a dense array. There has been considerable work on deletion maps, so constructing the set of all deletions in genomes. In 2006, there were concurrent papers in Nature Genetics. McCarroll et al. Presented a heuristic statistical approach to find clusters of Mendelian inheritance errors, null genotypes, and deviations from Hardy-Weinberg equilibrium. And these three conditions would suggest that there was a deletion in that set of SNPs. Conrad et al. In 2006 presented a more combinatorial approach where they were looking for, decompose their data into specific Mendelian inheritance patterns, and they looked for 2 or more Mendelian inheritance errors in a single trio. And that would suggest hemizygosity. Other methods since then have used different experimental techniques, tiling arrays, sequence data, like in the 1000 Genomes project, and arrays designed specifically to type structural variation. And example of this is the AFI 6.0. But it's important to note that these are all orthogonal analyses. So you can run these types of experiments and also use a SNP-based deletion calling method, as long as you have some sort of pedigree structure for your input data. So from either the SNP array or the next generation sequencing SNP calls, we convert the raw data into an intermediate representation, which we call a 0, 1, 2, 3 genotype. But it's very simple. It's just 0 and 1 for the homozygous major and minor alleles. 2 represents the heterozygote, and 3 is missing data. From this intermediate representation, we then convert it to a deletion vector, and this is in the alphabet of 0, 1, and X. 1 corresponds to an evidence of deletion, and these patterns strongly suggest that there's a deletion passed from parent to child. An example of this is when the parent and child are homozygous for different alleles. The only way to explain this phenomenon is by introducing a deletion or a genotyping error. We have consistent with the deletion patterns, and this example here is when both parent and child are homozygous for the same allele. You can explain this Mendelian inheritance just by normal Mendelian inheritance, or you can introduce a deletion. But it doesn't provide strong evidence for there being a deletion at the SNP site. And then no deletion, and these patterns indicate that there is no deletion at the site. This example is, it doesn't really matter what the parent is. If the child is heterozygous, then there's no deletion here. Both haplotypes are being seen by the experimental methods. So it's important to note that this conversion from genotypes to deletion vectors is essentially the same for inherited deletions that Conrad suggested in the 2006 paper. There's a one-to-many mapping here from evidence of deletion to category in A and B, because technically you can have an evidence of deletion with your mother or with your father. But the method isn't limited to trios. But if you do have trios, then you have these additional categories. So the deletion matrix is just the set of all deletion vectors. And we introduced in our previous paper this notion of compatibility between evidence of deletion sites. So 2 1s or evidence of deletion sites are compatible if there does not exist an X between them in either trio. So this represents sites that can be part of a similar deletion phenomenon. What I mean by this is the deletions could be shared by descent, or they could be independent recurrent deletions that just happen to overlap. And in this example down here with these 2 deletion vectors, this boxed region corresponds to strong evidence for there being a deletion because there's a lot of compatible evidence of deletion sites. We then model this problem with an associated graph where we have a vertex for every 1 and an edge between 2 1s that are compatible. And in this graph, cliques correspond to potential deletions. If this was your deletion matrix, this would be your deletion graph, and we would likely call this 3-clique a deletion and the isolated vertex a genotyping error. So this is really nice. We have a model to represent possible deleted sites. But any algorithm that's going to operate on this graph needs to be very fast and efficient. For example, here's a section of 774 SNPs in chromosome 22 of our autism data. And what we have here is every pixel denotes a different Mendelian inheritance pattern. So the black pixels are a combination of no deletion sites and consistent sites, so there's probably no deletion here. But the white and the red are potential deletions, where the red pixels are evidence of deletion sites. So you get regions of very complex deletion patterns. You also need an algorithm that's exact and doesn't miss any subtle signal. So you can imagine that within this deleted region, you might have a subset of your patients who have some deletion that's associated with autism. But if you were to just cluster this, and for example, and say that everybody in this interval has a deletion, you might miss that subtle association signal. So the formulation that we came up with for this graph is for each connected component in the deletion graph. And for each maximal clique in that connected component, we call it deleted if it's just above some threshold. And after we've called all the deletions, we report any evidence of deletion site that wasn't included in at least one deletion, a genotyping error. So in this example, we have two 5 cliques, two 5 maximal cliques that share a 3 clique. We would call both of the 5 cliques deletion if the threshold is 4 or less. So one of the results that we present in our paper is that there is this correspondence between maximal cliques and intervals. So each maximal clique uniquely defines an interval in the deletion matrix. And this is just an example where we have 4 maximal cliques and 4 intervals. So this is actually really nice because we know we have a polynomial number of maximal cliques in the deletion matrix. Therefore, we have a polynomial number of intervals. So we have a polynomial number of maximal cliques in the deletion graph. And in the 70s, Tsukiyama presented an output-sensitive algorithm, so an algorithm that could compute all maximal cliques in polynomial time per maximal clique generated. So in theory, we already have a polynomial algorithm to compute all of the maximal cliques in our data, and therefore all of the deletions. In practice, however, we implement the Braun-Kerbosch algorithm because it's widely considered the fastest maximal clique generating algorithm, especially for the type of data that we have that's very sparse. So the image I showed before isn't representative of the entire data set. It's in general very sparse. And sparse in terms of the number of evidence of deletion sites that cluster. So the software package that implements this algorithm is called DELISHUS, which is an algorithm for finding deletions in shared haplotypes using SNPs. Here's a sample of just the runtimes and the memory usage for some rather large data sets. So HapMap phase 1, European, HapMap phase 2 and 3, European, which, well, the phase 1 is the same data that was used in the 2006 papers. So we finished in under 2 minutes. And also the Aguiar autism data, which is 4,327 individuals and over 500,000 SNPs. We finished in just over 2 minutes, the maximum memory requirement of 2.5 gigabytes. I should say that this was run on a cluster, so this is the maximum requirement of any particular node. So the first thing that you have to do is tune your threshold parameter because you don't want too many false positives if it's set too low. What you can do, and you have to do this for each data set, is first calculate the frequency of the evidence of deletion consistent with a deletion, no deletion sites. And then you can generate a deletion matrix that's the size of one of the chromosomes of your data. So in this case, we're modeling it after the Aguiar data, the autism data. We have 25,000 trio inheritance patterns that we sample randomly from the frequencies. And we have 2,500 trios. And after we have this deletion matrix, we run the algorithm, and any deletion that's called within this matrix is obviously a false positive. The single individual algorithm here refers to just an algorithm calling deletions from some pedigree to a single affected child. So this is what Conrad et al. Was doing, where they were looking at one trio in isolation. So we see that we need a threshold of about 5 or 6 for the DELISHUS algorithm, and the single individual algorithm can get away with a threshold of 2 or 3 to have a tolerable number of false positives. After you define your threshold, you can do power simulations. So here we just take the actual autism data. we select a random interval defined, the size is defined by the x-axis. And then we change the genotypes to indicate that there's a deletion passed from parent to child. And this might not even introduce any evidence of deletion sites. It depends on the frequency of the alleles. But in many cases it does, and obviously the longer the deletion, the higher the probability that you'll introduce evidence of deletion sites. We see that for 5 individuals now, DELISHUS does have increased power over the single individual algorithm to infer deletions. Now, what we want to find is we want to find recurrent deletions. So we increase the number of individuals that have that are past deletions 2 from 5 to 25. So it's still relatively small considering the matrix is 4,300 individuals. But just with this marginal increase, we see that the power increases drastically, especially for smaller deletions. So it'll eventually flatten out when the deletions are big enough. But for small deletions, this algorithm does really well because it exploits information of multiple individuals at the same time. So one of the outputs of this software also is producing a deletion map if you're going to run this chromosome-wide or genome-wide. And here, the intensity of the violet color corresponds to the density of the deletions. And down here is that section of chromosome 22 that we were seeing before in the deletion heat map. With a slight modification to our model, we can actually infer de novo deletions. So if we have a de novo deletion in individual B, and this is, remember, the genotype representation where 0 and 1 were homozygous, 2 heterozygous, 3 missing data. If individual B has a de novo deletion, then whenever he's heterozygous, there can't be a deletion site. But now individual A is allowed to be heterozygous. So these are consistent with there being a deletion. So essentially, the probability of consistent site goes up. So this could be concerning if it goes up too high. You would connect the deletion graph, and you would get a lot more deletions called, potentially more false positives. So we ran the same experiment where we simulated these trio inheritance patterns to create a deletion matrix. And actually, the false positive rate does not increase too much. We can still use a threshold of 5 or 6 using the de novo DELISHUS algorithm. And just an example of what this effect has, this is one of the genes of interest in the autism data with the gene name replaced by X due to data usage rules. But before we had the second pair, and remember this is just red and white correspond to potential deletions, red being the evidence of deletion site. The second pair looks like he or she may have 3 separate inherited deletions. When we interpret this data using the de novo framework, however, this second pair becomes consistent and indicates 1 de novo deletion there. And if we see the effect on the deletion graph, it becomes much more connected. And we actually have a 5-click in here, which would be detected by the DELISHUS algorithm if the threshold was 5. So finally, we just present this result on critical regions. So of great interest in some autism studies is finding these critical regions, especially if you have those complex deletions that I showed before. So they're regions of maximal overlap among a set of deletions. So the problem statement is, given a set of deleted intervals now, find all of the critically deleted regions that include more than K trios. So the key observation here is you can construct a graph such that you have a vertex for each deletion and an edge between overlapping deletions. This vertex is chordal or triangulated, which basically just means we can construct this perfect elimination ordering, which allows us to find all maximal cliques really, really fast. So just to summarize, I've presented an algorithm for computing all potential deletions in SNP data with some sort of pedigree structure that's known a priori. It's efficient, and it extends genome-wide, so you can apply this to GWAS data. It's guaranteed to find all cliques, all maximal cliques, and therefore all potential configurations of deletions in your data. It can work in either an inherited or a de novo interpretation. And we also presented this critical regions algorithm. So DELISHUS is available at the Israel website, and I want to stress an alpha version. If you have any questions regarding on how to run it, or if you'd like to collaborate, then you can please drop me an email at derek_aguiar@brown.edu. We're continuously trying to, upgrade the documentation and to provide more of the features that were used in this paper to the general public. And it is a Java implementation, so it'll be easy to run on any machine. I guess before this, I finally want to say that, so there is current work for validating some of our top deletion calls, and if you want to talk offline about how we actually compute statistics for these inherited deletions, I would love to do that. But there is current work to validate those experimentally. Finally, I just want to acknowledge, first of all, the NSF Biodirectorate for providing me with travel funds so I could come here and present to you. My advisor and co-author, Soren Istriel, Bjarni Halderson, and Eric Morrow, also co-authors, and the various funding agencies. And thank you for your attention.
Terrific. Thank you for an excellent talk. We have 5 minutes or so for questions. I'll ask them.
Okay, cool.
And I'll ask it with a microphone so people can hear me. Read depth is also a way to try to infer something about deletions or even insertions, so duplications of regions. Have you tried to do any integrated analysis of read depth, say across an exon or a gene, and the SNPs that have been called for it?
So we haven't yet. So all of these other methods provide orthogonal analyses, so you can run them all at the same time. The problem with that is the Aguiar Autism data, they actually did not have any sequence data. We have a new data set from the Simons Foundation where they are actually starting to sequence. So you can do kind of a joint analysis where you look at the sequence data and you also look to verify that the SNP data is also concordant with the sequence data. But we haven't yet looked into doing that.
I have a question regarding your clique covering, or what it is. I didn't understand it. Did you want to make the cliques disjoint and you just couldn't do it, and so you accepted that the cliques could be overlapping, or is that not a problem?
So it's not a problem. If you have deletions that are shared evolutionarily, so by descent, then they should all overlap, and they should produce this maximal clique that's very easy to interpret. I guess all that point was you could have recurrent deletions that are independent, especially in regions that are flanked by homologous regions or duplications. You can have these deletions that occur independently, and they can overlap. And if they do overlap, we'll be able to detect the different intervals at which they overlap. So a set of deletions, even though you might consider them biologically to be all relevant, we'll be able to detect all of the possible configurations of that set of deletions.
Thanks.
Yes, I have a question about your power graph. It looks like it plateaus before 1. I was wondering if that's some inherent assumption there that you cannot get to 1.
Is this the one?
No, the power graph. The power graph.
Oh, power graph.
It looks like they never reach 1.
Oh, it never reaches 1. Okay, yeah. So right here, we're most interested in finding small recurrent deletions because these are the ones that often get neglected or not analyzed enough. If you extend the x-axis out, you'll get much more SNPs covered. And what you'll see is all of the methods converge, and they'll converge at 1. So if you have enough SNPs, you're going to find the deletion. But to us, we want to find the small deletions. So I just want to show that we have superior power than the other algorithms for the smaller deletions.
So I was just curious, what would be that number? And also, and also if this is very sparse data because it's from SNP arrays, right? And I was curious to see if you tried to do sequencing data, which is going to be more dense and perhaps a little bit more noisy.
So first, I took this out at the last minute because I wanted to be quick. But here is a power analysis where we're simulating deletions in base pairs now. So we might not even actually hit a SNP. I guess this is more how it would happen biologically. But if we extend this out, it looks like they converge. They converge around 50.
4,000?
Yeah, about 40 or 50,000 or so. And to your second question, I would love to apply this to sequencing data. It's just the case that these association studies they can't sequence this many individuals and their parents. So we don't have access to any sequence data that's a whole genome for many individuals.
So one more question. Maybe you could go forward to your deletion in autism. So, oops, backwards.
Oh.
One more. That one. So chromosome 17, 16, 8, and 9 seem to have some remarkable recurrences of regions that have deletions. Have your collaborators drilled down on these and found specific genes in those regions that make sense, or is there some interesting inferences that people are already making about this data?
So, well, this image, first of all, it's cut off, so it extends much farther. Okay. But there are these known regions of complexity throughout the genome, and a lot of them have been associated to diseases like autism. But the thing is, they're very complex, so what people do is they just say, this is a region that may affect, but you can't really say much about it unless you detangle the different types of deletions. So we actually don't find too much in those because they're usually common deletions. And very common deletions you wouldn't expect to be associated to any disease. But it's still a good question, and you still should parse out these regions and see if you can find anything really.
Yeah, and I guess how many deletions per person are you able to infer?
So I didn't want to show any statistics like that because we haven't gone back to the DNA yet and validated these. But we do find a lot of sites that seem to be true deletions, which is it's quite exciting actually, and it just shows you that you really need to have the full spectrum of variation in order to say a convincing statement about what's associated to autism disease.
Thank you for a really exciting talk.
Thank you.